That's off.
University Medical Center. He develops and applies innovative mass spectrometry-based phosphoproteomics methods and data analysis strategies to improve early diagnostics and treatment of cancer. He is responsible for the Q executive LC-MS infrastructure and focuses on protein modification and signal transduction. Now, before we move on to Dr. Piersma's talk, please allow me to introduce all the panelists. The first panelist is Professor Yang Bian. Professor Yang Bian is from the College of Life Science of Northwest University, China. His research focuses on the development of new methods for PTMs and high throughput proteomics. He develops the SH2 superbinder tool for tyrosine phosphoproteome analysis and microflow LC-MS, enabling the analysis of nearly 40,000 samples within 2 years. Next, Dr. Zhenpeng Zhang is a postdoc researcher from Beijing Proteome Research Center. Dr. Zhang is involved in the study of tumor precision therapy based on proteomics and post-translational modification omics. Its main research interests include screening tumor neoantigens by proteome genomics technology and developing personalized immunotherapy, predicting tumor tissue-specific aberrant activated kinases by phosphoproteomics technology to screen targeted kinase inhibitors and guide clinical treatment. In the past 3 years, he has published 5 articles as the first or corresponding author. And Dr. Zhang Cheng is a postdoc researcher at Guomei's lab of Westlake University. He received his bachelor degree in applied chemistry and a PhD in chemical biology from Fudan University. During the doctorate study, his research mainly focused on developing new methods and technologies for the post-translational proteome analysis, such as phosphorylation and glycosylation as nitrosylation. He was an R&D scientist for new drug discovery before joining Guomics in April 2021. He is now embarking on a new journey in clinical proteomics research. We also have Meng Ge with us today. Meng Ge received clinical medicine and biomedical science training in the joint program of Nanchang University and Queen Mary University of London. While well trained through scientific research projects in the laboratory of Baoming Li and Xu Huizeng of Nanchang University. She also went to the laboratory of Yi Rao at Beijing University and the laboratory of Benjamin Stiglitz at the Queen Mary University of London for summer internships in 2016 and 2017 respectively. She is now a PhD student at the Guomics Lab of Westlake University, working on protein complex and pan-cancer proteome research. Without further ado, let's welcome Dr. Sander Piersma to share his talk. Dr. Piersma, the floor is yours, please.
Okay, thank you for the very kind introduction, and I'm impressed by actually the panelists. So I really look forward to discussion after the presentation, and I guess we'll keep questions until the discussion part at the end. So it's good. So I'll share my screen and see whether everything works. Yes, I think this is okay. Okay, so indeed, today's topic is kinase activity inference, specifically from phosphoproteomics data. As you can see here at the right side, here is the building that we are in. The presentation that I will be showing today is mainly based on a review that we wrote last year, and it's published this year in mass spectrometry reviews. And that I will start with that to have an outline on what kind of tools we use for kinase activity inference and why we do it. And I will then continue with our own favorite tool, the one that we developed. And I will show a preclinical application where we used kinase activity inference to find combination treatment targets. And the papers on which this presentation is based are to be found here. So this is the review, and this is the tool that we developed, description of the tool. And here we have the application of the tool in the context of TALL, and I will introduce that subject later on. I think I will be taking about half an hour to 45 minutes, but well, let's see. So I have to move my screen a bit so I cannot see the screen. Yep.
Yes, we can see the screen.
Yeah, but I could not see my own slides because the camera was blocking it. Okay, so why would we want to do? Yeah, do we want to do kinase activity inference, and why is it of relevance in cancer research? Well, the rationale is that many kinases are oncogenes. So many kinases have acquired mutations that are activating, and this is actually one of the causes of the cancer. At the same time, the pharmaceutical industry has spent a lot of time and effort on developing kinase inhibitors. So kinase inhibitors are the largest group of targets currently in research in pharmaceutical industry. So we both have oncogenes on one side that are kinases that are uncontrolled, and then we have a lot of small molecule and also antibody-based drugs that can actually inhibit kinases. So in the human proteome or human genome, there are 530 or slightly more protein kinases. And currently, I think that's already outdated. There are like 30 approved kinase inhibitors in the clinic. In the pipeline, there's at least an order of magnitude more kinase inhibitors. So in the coming years, many more kinase inhibitors will enter the clinic. And one of the apparent drawbacks that we see in clinical practice is that only a subset of genetically selected patients, so that means patients that have a oncogenic mutation which is recognized, which we can detect by genome sequencing. Even if we take those patients and give them the drug that should actually inhibit the aberrant kinase, then still only a fraction of those patients will respond. In addition to that, also resistance development is an issue with kinase inhibitors. So there are a lot of reasons to focus on kinases. There are many drugs that are actually targeting kinases. And at the same time, we're not very good at getting the right kinase inhibitor to the right patient. So that's really a clinical problem that needs to be assessed and solved. The key tool that we'll be talking about today is kinase substrate relationships. So for every kinase, we would like to know which proteins is actually their downstream target for phosphorylation. And there are such databases available. Phosphocyte Plus is most likely the best one, but also PhosphoElm and Swissprot and HPRD and Senior are great sources for finding kinase substrate relationships. And many of these databases actually are updating each other. So in the end, we use most of the time Phosphocyte Plus, but also the other databases have their merit. So phosphocytes, so parts of proteins that are phosphorylated, are the product, by definition, of an upstream kinase activity. So for every phosphocyte on a target protein, one or more kinases have been active. And most of the time, that sequence context dependent. So for every phosphocyte with a specific sequence context, there will be a different kinase responsible for its phosphorylation. So this goes away from the one gene, one protein idea. Every phosphocyte is independent and has an upstream kinase or multiple upstream kinases. So the limitation currently is that kinase substrate relationship databases are far from complete. So for many phosphocytes, we don't know what the upstream kinase activity is. Conversely, for several kinases, we don't know what their actually sequence preference is. So we don't know which phosphocytes are actually produced by the kinase. This is really a limitation, but at the same time, it doesn't mean that kinase substrate relationship databases are not useful. So they can be used because heavily studied kinases and heavily studied phosphocytes are contained in these kinase substrate databases, and therefore that data can really be used. Phosphocyte annotation, the functional annotation of phosphocytes, remains a challenge. So there are many, many more phosphocytes detected currently reported in literature than there are annotations for those phosphocytes. So for many phosphocytes, we actually don't know what the physiological role is, if any. Okay, so kinase activity inference is a method to use the phosphocyte footprint, so which phosphocytes at a global level are phosphorylated, to predict the relative kinase activities of the relative upstream kinase activities. So we start from phosphoproteomics data. So we start from a very comprehensive large set of phosphocytes, and then we try to infer which kinases have been active. And by adding up the activities, adding up the observations of the phosphorylated substrates, we infer the activity of the upstream kinases. Okay, so I now go one step back, and for many of you, this will be information you already know. But for the ones that are more or less new to the field, I will first give a brief introduction on phosphoproteomics. So phosphoproteomics relates to phosphorylated proteins. About 30%, or probably a little bit more, of the human proteins are phosphorylated. Phosphorylation is a reversible covalent modification. So a modification with a single phosphate group onto the onto 3 amino acids out of the 20 natural amino acids, and the primary activity that catalyzes this phosphorylation is a kinase in the presence of ATP. Many cellular processes are regulated by phosphorylation, and it's the key step in intracellular signal transduction. So receptor signaling is relayed through the nucleus to start different gene expression programs by way of phosphorylation and dephosphorylation. The 3 key amino acids that can be phosphorylated in nature are phosphoserine, phosphothreonine, and phosphotyrosine. And this is the proportion in which the phosphorylation occurs in nature. So the majority, more than 85%, is on serine, about 12% on threonine, and 1% or even less on tyrosine. And that's a bit of a drawback, because tyrosine phosphorylation is one of the most important, or at least one of the most interesting phosphorylations, but it's only a minority. In the next slide, I will show you why that it can be a bit of an issue. So we are looking at a reversible reaction. Here I only show the kinase reaction, but there's also, of course, also a phosphatase that will dephosphorylate phosphosites. So there's a dual level of regulation. Kinases catalyze the reaction, the forward reaction, and phosphorylation, and phosphatases catalyze the reverse reaction, dephosphorylation. And we are a proteomics and mass spectrometry group, so obviously phosphorylation is accompanied by a big mass shift, and therefore we can use mass spectrometer to actually follow phosphorylation on a global scale. So if you talk about phosphorylation, you talk about phosphopeptide enrichment. Why? Well, phosphosites tend to be substoichiometric, so they are of lower abundance than non-phosphorylated peptides. Phosphopeptides show, in general, a lower ionization efficiency compared to normal peptides. And when doing database searching, so the way to analyze mass spectrometry data and actually identify phosphosites, these tools result in lower MSMS scores, identification scores for phosphopeptides. So if you have a mixture of phosphopeptides and non-phosphorylated peptides, the great abundance of your signal will be non-phosphorylated. The way to actually zoom into the phosphorylation is to enrich for phosphopeptides. So there are 2 general methods: global phosphorylation enrichment, where the method doesn't have a preference for the serine, threonine, or tyrosine phosphorylation, and more selective methods where we zoom in on phosphotyrosine only. So titanium oxide, we've used that for a few years. It's a very convenient enrichment method. It has a high yield, but you get acidic peptides as that co-isolate with phosphopeptides. And then currently we are using IMEC on a Bravo robot to do global phosphorylation enrichment with iron NTA stationary phase. And that's a very beautiful method to do global enrichment, a very convenient method. For phosphotyrosine, we use the phosphotyrosine specific antibody from cell signaling, but there are others around, including the SH2 domain material that one of the panelists is actually using and pioneering. This is a method to select only for the 1% of phosphotyrosine enriched phosphotyrosine peptides, and we don't capture the 99% serine threonine phosphorylated peptides. A small issue about phosphoproteomics, it's harder than normal expression proteomics. Why is it harder? Well, you have to enrich, but once we've done that, it's still a little bit harder because we're not looking just at a amino acid sequence, which for identification and quantification would be the end stage for a data-dependent MS experiment. Get a list of identified peptides, and that's it. For phosphoproteomics, we have serines, threonines, and tyrosines that can be phosphorylated, and we know by looking at the delta masses how many phosphosides or how many phosphorylations there are on the peptide, but that's still what remains is the question, which of the target amino acids is actually phosphorylated? And it means that you have to go into the fragmentation spectrum to find the site localization diagnostic ions. And fortunately, software allows you to do that and gives a probability score, which of the threonine or tyrosine in this sequence example is actually phosphorylated. And depending on which of the fragment ions shows the mass shift, we can infer which of the amino acids has actually been phosphorylated and what the probability is that it's phosphorylated. There are different tools around that can do that. So we normally use MaxQuant for DDA data, and for DRA we use Spectronaut or Diane. And it will give you phosphosite identification and quantification and localization. Those are the three levels of information that are further used in downstream analysis. Okay, so now I go to figure 1 of the review, which summarizes part of what I just told you and extends it to what I will be talking about. So we start from biological material, tissues and cells. These cells and tissues have experienced kinase activity just because of proliferation of the cells. So this is not an in vitro part. This is just what happens in the cells. And once we harvest cells or tissues, then we digest it by trypsin into peptides. From the peptides, we do phosphopeptide enrichment, either global or PTAR selective. And then we do mass spectrometry, typically 1.5 hour, 2 hours reverse phase separation coupled to a Q-exactive mass spectrometer. This will give us 20,000, 30,000 MSMS spectra identifications, and this will result in typically about 10,000 phosphosites. Depending on your mass spectrometer, an LC system, you can do this in an amount of time that you allow per experiment. Anywhere between 5,000 and 30,000 phosphosites can be observed in a single experiment in a single sample. So once we have a data matrix for all our samples containing phosphosites, so sequence and phosphosite localization, and we have a quantitative value, either intensity-based, based on MS1, or spectral counts, or LFQs, also based on MS1, a quantitative value for each phosphosite. Then we can apply downstream analysis tools to infer from this complex collection of phosphosites and relative abundances. We can start inferring which upstream kinases have been active. So the three main tools that I will also discuss later on are kinase substrate enrichment analysis, KC, which relies on mapping all of the observed phosphosites to their upstream kinases. And it of course depends on the completeness of the database, as I already indicated. And typically this is a method that's based on a fold change. So you have a differential condition, cell line plus or minus stimulus or plus or minus inhibitor. And then we look at the behavior of each of the phosphosites and map these phosphosites to their upstream kinases, and therefore thereby infer kinase activity or a kinase activity change depending on the condition. So that's KC. This was a method that was initiated or developed by Pedro Cotillas in the UK, and is probably the start of the field. And there's different implementations of this tool available with different names, but under the hood, they all do the same thing. I will show you later on what it actually, what the key algorithms are. Then we also have PTM substrate enrichment analysis, PTMC, and this is based on the MSigDB, PTM-SigDB, and this is a curated database which maps phosphosites to biological pathways. So it's a very integrative analysis, and what you actually do is like a gene set enrichment, you calculate an enrichment score. The third method is called INCA. That's our own tool where we focus not only on phosphosite kinase relationships, but we also zoom in on the kinase itself, because it turns out that kinases are themselves phosphorylated, and kinase phosphorylation is a key indicator of kinase activity. So we integrate the kinase substrate relationship with kinase phosphorylation and integrate it into a single score. And I will show you that you can use this score to find drug targets. So this is in a nutshell what kinase activity inference is. Many tools have been developed in the past 10 years. It's quite an active field. Many of the tools share the same basic ideas. It's either single sample or group-based analysis, and it's a way to map phosphosites as we can measure them easily to upstream kinase activities, and then sometimes enrich that with protein-protein interactions or with pathway information. Some of the tools are made for a single application. Other tools are made as a tool for the community to use. And you can see with the dates that it's really quite active. I will not go into detail about all of them, only KC app, INCA, and PTMC. But in the review, you can read short descriptions of each of the tools and their history and when they were actually implemented and with which examples. So one of the things we did was a small bibliometric analysis. So we searched all of the tools, and then what we looked at is in the ICI Web of Science, and what we looked at is the number of citations here on the y-axis. Here, obviously, the date of introduction to the tool, publication year, but also here the size of the bubble plot reflects the number of active users. And you would think that older tools are used more and are cited more than new tools. Well, that's to some extent the case. But what you can also appreciate is that there are tools out there like P-chips, for instance, that are cited many, many times, but there are actually not that many users. So what we did was we not only look at the citations, but we also looked into each paper, and if a tool was actually mentioned in the materials and methods or in the results section, that is a proxy for actual use of the tool. Whereas just citation, mentioning it in the introduction or discussion of paper, usually is more like a general statement. You can infer kinase activities from the phosphosite footprint without actually performing that. So here we want to zoom in on the actual users. So how often are tools used? And then you see that the most popular app, the most popular tool is the KC app, followed by PTMC and INCA. And that's also the reason why we zoomed in a little bit more on these three tools, because they have been used and applied most often compared to the other tools. And you can also see that also last year when we were writing, many new tools popped up, which integrated part of the performance of the older tools, but of course they have not been used by others yet. So in the coming years, these bubble plots will change, and multiple tools will actually develop, integrating different levels of information. So protein-protein interaction, functional neighborhood, that's ROCI, for instance, more signaling pathways like PTMC. So I think there's a lot to be expected in the near future. So the key algorithms, and I'm not sure whether I go really in detail or just show you the overview, but for the three most popular tools, KC-app, PTMC, and Inca, KC-app is actually, the formula here is a z-score transformation, and it actually treats the fold change of substrate phosphosites for a specific kinase and compares that to the mean fold change over all phosphosites. And then it also uses the metric, the number of phosphosites that actually are identified for this specific kinase, and the standard deviation across all kinases, or across all phosphosites. So this z-score gives you a score that can be used to compare upstream kinase activities. And for all of these tools, holds that the power is the actual integration or the aggregation of individual phosphosite intensities to a kinase, to attribute multiple intensities to a kinase. And with this quantitative value, you can actually, in differential conditions, tell something about the relative kinase activity or the change in kinase activity. And that's a thing that you can really use. KC is by definition a two-group comparison. PTMC and Inca can be used as two-group comparison tools, but also as single sample tools, depending a bit on how you use the algorithm. So for PTMC, we have a PTM-SIGDB. So we have a rank sorted list of genes, or actually phosphosites, and we can calculate an enrichment score. So this enrichment, that's the key thing behind PTMC. Inca works a little bit differently. For Inca, you can see as two different sides of the same activity. So we have four components, two components that are related to kinase phosphorylation, and two components that are related to substrate phosphorylation. And then the combined Inca score is the geometric mean of the kinase component and the substrate component. So we use Inca mainly as single sample analysis tool, but obviously if you have two groups, you can group Inca scores and do a statistical test. And then you have a two-group comparison form of the same tool. So all of these tools, one way or the other, use phosphosites and map them either to individual kinases or to pathways involving kinases. For all the other tools that we described, similar variations can be found. This is the core algorithm, which is also part of Inca, for instance, is this part. And it's also present here. So it's not independent or completely different algorithms. They are all informed by similar concepts, but then the implementation and statistics and the additional data streams will differ between tools. Okay, so the next part of the review was actually a comparison of different tools, hands-on. So one of the postdocs in our lab, Frank, did an experiment in which he used U2OS cells and either did not treat them or he irradiates them. So what you do by irradiation is induce DNA damage. So what we expect, and what we can also measure, and what also others have measured before us, is a DNA damage response. And that's typically ATM, ATR, in check 1, check 2, signaling goes up. So we did this experiment, and we've also, in a different context, we have published this in MCP. And we used the MAX1 search and used that as an input for PTMC, the KC app, and Inca. And what you can appreciate is that you get a ranked list of kinase activities. And what you can also see is that for instance, KC as well as Inca produce lists of individual kinases, kinase names, and a enrichment or a score value. And this is all a two-group comparison, so non-treated versus irradiated. But for PTMC, you can see that the names are different. We are not looking at single kinases, but we are looking at pathways or signaling pathways that are enriched. So we're not zooming in to a single kinase, but it's more a collection of pathways that share a biological, a collection of kinases that share a biological pathway. And so here already the protein-protein interaction network is more or less integrated into the database. So that has its merits. You can also see that different statistical qualities are included. So here, these two have FDR control. Here we have, you can calculate an adjusted p-value. So the tools are similar, but definitely not identical. But when looking at the main differential processes, we recover more or less the same things. So all three tools will find back ATM, ATR, check2, upregulation, or increased activity. So in that sense, they don't differ that much. But if you look more in the details, so then you see that there are individual kinases that are missed or that are found by specific tools. And whether that's real or that's noise, we actually don't really know. We know that at the top of the list, so the top here, each tool recovers the same type of signaling, the same ATM, ATR check, and that is reassuring. But obviously the more detailed things at the bottom of the list remain to be validated. Okay, so here a qualitative comparison of the different tools. So KC app, you can either use the web tool or an R package. With PTMC, you use the web tool via gene pattern, or you can download also an R package. And for Inca, for all outside users, the web tool is available, and in our lab we also have the R version locally. KC app is by definition two-group comparison. PTMC can be used as two-group and single sample comparison. Inca is actually made for single sample comparison, but if you have grouped data, you can do a post hoc group-based analysis. Inputs are a CSV file with the canonical UniProt IDs for the KC app, a GCT file with phosphosite with flanking sequence, or the UniProt ID and the phosphosite plus site group ID, and for Inca, the MAX1 output text folder. So the output is a list of kinases for the KC app, as well as for Inca, and then Inca is also enriched with a kinase substrate relationship network. You get that in addition to the kinase ranking. And then for PTMC, you have kinases as well as other pathways. So kinases that are part of a pathway are reported, but is not exclusively single kinases. So the kinase substrate relationship database, there's quite a lot of overlap. Phosphocyte Plus and Networkin is used by KCApp and also by INCA. But then INCA is enriched with kinase-specific observations, such as activation loop phosphorylation. And PTMC also relies on Phosphocyte Plus, but also other pathway information sources that are actually inserted in a curated database. Kinase substrate relationships are not static, so this is a field that's actively being developed. So it makes sense to update or be able to update your KS kinase substrate relationship database when those databases are actually improved in time. So the KC app only allows the author to do that. And well, Frank wrote here that it's manually possible. I don't know, but that's really the case. PTMC is also by the author needs to do that, and for INCA that's the same. Currently, KC app is human only. PTMC is a little bit wider, and they're both human, but also mouse and rat are available. And currently, the current implementation of INCA is also human only, but we have a mouse version that's being developed, and it will be reported shortly. Okay, so I've now shown you many tools. I've zoomed a little bit in on the three mostly used tools, and now I'm going to zoom into INCA, our own tool, and spend a little bit of more time on what it does and how it performs. So, as I already told you, INCA is based on two sides. So we have the kinase components and we have substrate components. And database search in MaxQuant will give you phosphopeptides and phosphosites. So phosphosites can immediately be mapped to Phosphocyte Plus, so our kinase substrate relationships database. And but also, in addition to that, because it's not a very comprehensive database, we also use a sequence-based kinase activity prediction algorithm. So based on the sequence of the peptide, it can predict which kinase has been active. It's called Networkin, and it works similarly as Phosphocyte Plus, but then only based on sequence and not on really phosphocyte kinase relationships. For phosphopeptides, for all phosphopeptides that we detect, we look whether they are actually part of a human kinase, and if so, it will be scored separately. And for the activation loop, that's a specific part of the sequence in most human kinases that upon phosphorylation actually increase activity. So it's a proxy for catalytic kinase activity that's scored separately. So and what we typically use, we can use phosphocyte intensities based on the MS1 signal, but you can also use spectral counts, and that's what we historically have been using, and that works very well when you aggregate data. If you have many data points, spectral counting is very straightforward and works beautifully. So what you can see here is the overall equation where we have the kinase component and the substrate component, and so we add up spectral counts for all phosphopeptides that are part of a human kinase. We add up all spectral counts that map to the activation loop. That's this part here. And then for the phosphocyte, for the substrate part, we aggregate the spectral counts from the Phosphocyte Plus database and from Networkin. And what you can see here is we multiply and then take the square root. So we calculate the geometric mean. And it also means that if a kinase is observed only directly from its own phosphorylation, but not from its substrates, or only from its substrates, and there's no direct observation, then the INCA score will be 0. So therefore, INCA is a pretty strict kinase activity inference tool. Both the kinase as well as its substrates need to be detected in the dataset. We developed this based on phosphoproteomics experiments with known oncogenic driver cell lines. And then our observation was that when there is a clear oncogenic driver, so for instance, BCR-ABL here in K562, then BCR and ABL will be high ranking, the top 5 or the top 3 of the INCA scores. That was our first starting point for developing this further and testing the hypothesis that you can use direct kinase observations as well as substrate observations and integrate them into a single score to have a proxy kinase activity. Since we are working from kinase substrate relationship databases here, it's trivial to actually make for each analysis, make a KS plot in which you can see the more connections a kinase has, the stronger its activity actually is. Alex is responsible for the code, and we currently have quite a lot of registered and active users. So going beyond the 4 examples that we used in the primary paper, we now have many more cell lines with known oncogenic drivers, where we can use INCA to find the responsible or rediscover the driver kinase. And here you can see in color that the known driver kinase in color is in a clearly oncogenically driven cell line is typically in the top 4, top 5, but it should be really well visible. So this gives us reassurance that kinase is that INCA gives you a score that can be used for finding oncogenic drivers. Okay. So now to finish off with a real-world example, I will show you work of our PhD student, Valentina Cordo, who works in collaboration with the Pediatric Oncology Institute Maxima Center in Utrecht. And she worked on T-cell ALL, T-cell acute lymphoblastic leukemia, both cell lines as well as PDX models. And what we try to do is to do global phosphoproteomics, to find targetable kinases, and to find which combination treatment, so which combination of kinase inhibitors works best. Because what we already know from both clinical but also preclinical research is that single inhibitors often don't do the job. So T-ALL is an aberrant proliferation of immature T-cell progenitors and makes up about 15% of all pediatric ALL cases. So all children tumors. And we aim for identification of active kinases, and this provides a rationale for effective combination treatments. And so what we start with is a collection of T-ALL cell lines and also T-ALL patient samples that are then implanted into mice to make PDX models. So from both the cell lines as well as the PDX models, we isolate the phosphopeptides, both with phosphotyrosine IP, but also with global enrichment, do phosphoproteomics analysis, and use INCA to rank kinase activities. And globally, we reach a depth of about 20,000 phosphosites, 5,000 phosphoproteins, and we see about 200 phosphorylated kinases. In addition to this, we did in vitro drug testing. So for the cell lines that we applied, but also the PDXs, we made inhibitor titrations and looked at survival for single drug and multiple drug combinations. And what we saw is that inhibition of these cells are typically LCK driven, so dasatinib is a good drug. but we could also see that dasatinib only works in a subset. And you need another signaling axis to actually be blocked in order to really sensitize the TALL cells to dasatinib treatment. And typically, insulin receptor or IGF-1R receptor signaling is a very good sensitizer. Okay. So I will not show you any of the cell line work, but only the PDX as examples. And this also stresses the case that each tumor is different, and that there is no single recipe to find the right targets. So what you can see here is the INCA scores. So here we have the kinase name, and here we have the INCA score value for 4 different. I'll show you the 3, but the fourth one will be the next slide, with 4 PDX models. And what you can appreciate is that on the top we have LCK and SARC and FIN. So this is all SARC type of signaling, and dasatinib is a good SARC-enabled inhibitor. What you can also see is that there are also other kinases still active here. GSK, alpha and beta is typically in many experiments, it's more like a household kinase. But for instance, the insulin receptor here is a non-standard kinase that you don't observe that often. So when we saw the insulin receptor here at a prominent position, we speculated that might be a therapeutic target. You can also see it's not in every PDX. So for instance, PDX3 doesn't have any insulin axis signaling at all. So we have dasatinib, that's our key SARC-ABL inhibitor. And then we applied this BMS inhibitor, which is specific insulin receptor IGF-1R inhibitor. And we did combination treatment. So here we see mono treatment of with both dasatinib as well as BMS, and here there's a additive effect. But for instance, for PDX2, we see that the combination treatment is really synergistic. So it's better than the sum of the two. So when we see this insulin receptor activity, combination of dasatinib as well as insulin receptor can really inhibit cell survival. Both of the inhibitors by themselves don't do that. You really need the combined activity across both signaling axes. In PDX number 3, there is no target. The insulin receptor and IGF-1R is not found, activity is not found. And what we then see, if we do combination treatment or individual treatment, there's really no benefit at all. So cell survival is not really affected. So we can see here that the BMS drug that inhibits the insulin receptor and IGF-1R receptor can synergize with dasatinib, but only if those activities are actually present. So you need the activity plots to actually find this target. And here we have PDX3, but also PDX4. And in PDX4, there is a bit of insulin receptor activity, but that didn't really do much. But what we did see was highly prominent JAK activity, and JAK is also not often observed in INCA plots. So once we see this popping up, we know that's a really unusual activity to be there. And unfortunately, there is a drug, ruxolitinib, that's a pan-JAK inhibitor. And what we see here is the same as for the insulin receptor in the previous slide. So if the target is active, and we see the target being active because of the phosphosites and the kinase that we can find in a phosphoproteomic experiment, then the drug actually works, and it works also synergistically again with dasatinib. So we see only a benefit when the kinase is active, and that's really a big take-home message. It makes sense to look at which kinases are active, because it influences the selection of inhibitors. And if you would give this JAK inhibitor to a patient that does not have enhanced JAK activity, it really doesn't do anything. It's completely useless. So you really need to match the combination treatment to the kinase activity profiles. And I hope to have shown you that phosphoproteomics can give you a handle to actually do that type of analysis. So to summarize, kinase hyperactivity in cancer can be detected by INCA and other tools in a phosphoproteome-wide assay. We're not upfront zooming in to any specific kinases. We keep a completely open view, and we let the phosphoproteome tell us which kinases have been active and base our decisions on that. So what I showed you was ex vivo validation that selected drug combinations in PDX and organoids. I didn't show you organoids, but only PDX model that works. And what we see is, as a clinical future is that INCA or other tools that can use single samples. So group-based analysis doesn't work well with single patient samples. But once you can work with single patient samples, then what we see in the near future that we can produce a ranked list of actionable kinases, so kinases for which inhibitors are available. So this gives like a short list that the oncologist can use to help in treatment selection. And there will be still many steps before we go into clinical real time, but we are working very hard on achieving that. And with this, I want to thank you for your attention and take any questions.
Thank you, Dr. Piersma. Thank you for your excellent talk. And I think we can see the potential future of this software in the next clinical trial. And I think now let's go on to the panel discussion. First, I have a question. Just as slides as you showed, the phosphorylation site determination is based on the mass spectrometer data. Sometimes, not sometimes, we always get some phosphorylated peptide that we cannot exactly determine the phosphorylation site when there are some peptide contains more than 2 or 3 phosphorylation sites, but maybe there's only 1 phosphor group added onto it. So what do you do with these phosphopeptides? Just add them into your analysis list and annotate with a possibility, or just remove them, or do you do some further analysis?
Yeah, so that's an excellent question. So indeed, anyone who has done phosphoproteomics analysis will know or appreciate that there is uncertainty in the data, and especially phosphosite localization. And what we typically do is just to cut off. We only take class 1 phosphosites, so it means that the localization probability for each phosphorylation is 75% or higher. That's a bit arbitrary. It's been used already since Jesper also introduced that, I think almost a decade ago. It helps to clean up the data a bit by selecting only for confident localization sites, but whether that should be 75% or 80% or 90 or 95 or 99, I don't think it's that critical. And the reason why I think it's not critical, or not critical in the sense that you have to have like 100% localization probability, lies in the fact that for kinase activity inference, we aggregate the quantitative value for many phosphosites to its upstream kinase. And therefore, even if there is uncertainty or probabilistics at the individual phosphosite level going on, it will average out to some extent to the kinase level. So since we aggregate multiple data points to a single number, you can allow for noise, localization noise in the primary data. So it is good to filter for relevant localization probability. And I think the class 1 definition is okay for that. You also should be aware that localization probabilities can be much lower. So we sometimes see it's just 50% or less. And of course, that's not really useful, because if you have like very low localization probability and you keep all of that noise in your data set, then you start making many different connections to kinases that are really not correct. So it helps to filter, but you don't have to filter for 100% probability. There's like an optimum, and I think the 75% is okay-ish. But it's good to be aware.
Okay, thank you.
Yeah.
Thank you.
Yeah. Thank you for your talking. And I have one question. So I think the accuracy of the prediction is based on, we know the relationship of the protein kinase and the substrate size. So how do you get this information?
Yeah. So like I already told you in the presentation, there are different sources of kinase substrate relations. I think the best curated, most comprehensive database is Phosphocyte Plus, but it's not the only one. And Phosphocyte Plus is a very rich KS database, but at the same time, many of these phosphocytes are unannotated. So we don't know what the biological function is, and we also don't know what the upstream kinase is. So there's many more phosphocytes in Phosphocyte Plus than that there are phosphocyte kinase relationships, and that's I think an active field that really needs to be even more active than it is now. So to establish bona fide tissue-specific kinase-substrate relations, that's really important for the whole field of kinase activity inference, that these databases are getting richer, but also of higher quality. And there is no simple answer here. Yeah. So it can, I think the evidence level in Phosphocyte Plus is based on both in vivo as well as in vitro experiments, but there's no single experiment that will qualify a kinase substrate relationship. So I think there's a lot of work still to be done there.
Yeah, I think there are, in the Phosphocyte Plus database, you have different levels of data. So like you say, in vitro, in vivo, and also mass spec side data. And but I think maybe you can also try the kinase substrate motif. So this will maybe let you have more substrate and kinase data points, and then you can make full use of your data. Because now if your reference is bigger and you can have more data.
This was actually a very good point. This was actually the reason for us in the development of INCA to not only use Phosphocyte Plus, but also use NetworkIn, which is a motif prediction, kinase activity prediction tool. And what we saw is that NetworkIn works nicely, but you have to really control it very tightly, because if you don't put the cutoffs high enough, it starts to generate all kinds of nonsense motif predictions. So we're quite strict, and it's also what the primary author Rune Lindling described, I think, in the last version of the tool, that you should be aware of the thresholds that you need to apply to have a proper prediction. And once the thresholds are high, the prediction space actually narrows down already a bit. But it's a good way to get kinase substrate relationships in the absence of a Phosphocyte Plus entry. So you can use both, but you need to take some care that you are not predicting too much.
Yeah. So
yeah.
I think if you will use the kinase substrate motif information, maybe we can also introduce the protein-protein interaction network and some other database. So at least the protein and the kinase should be somehow in the same place, or they have some kind of interaction.
Yeah. So they should actually meet each other in the cell. So protein-protein interaction is a very good criterion for inclusion. So if a protein has never been identified, the kinase has never been identified as an interactor with its substrate, it's very unlikely that's a really in vivo substrate. So indeed, different information levels need to be integrated. And that's also what you see in multiple of the tools that I did not describe, that actually protein-protein interactions, but also for in Rokai, functional, what was it now? There's a very nice term for that. Functional context. So phosphosites that are part of similar biological processes are most likely to be real interactors. Well, I'm not sure whether I'm formulating it right, but integrating multiple levels of evidence is really important, and mapping to pathways, but also integrating with multi-omics. So having RNA-seq and genomic information can already also help. So it's I think the data integration part, that's where the real future will be, so that you integrate phosphosites and kinases, but also protein expression, protein-protein interactions, and signaling pathways, of which so much is known already, and to do that in a comprehensive way. Yeah, so I think the bioinformaticians still have a lot of work to do.
Yeah. So you show some very exciting results on your xenograph data. So I have one question about the data. Has these xenographs from the same patient? Because you have at least 5 xenographs, and they show somehow a little different pattern. So are these from the same patient?
No, so they're different patients, obviously. Yeah. And what you can see is that all, and that is also with the cell lines of these TALL cells, they all, if you normally we only look at like the top scoring kinase activities, and they are all the same. So we have, I think, 15 different cell lines. They all have LCK, SARC, LIN, FIN in the top. And then you think, okay, then the dasatinib would be a good pan-SARC inhibitor, which also has an ABL activity. So that should inhibit all of cell growth in all of these models, and it will only do that in 2 or 3 examples. And in the PDX, that's more or less recapitulated. It's also, they all have the same top activities, but only in a few cases, dasatinib will be sufficient. Actually, there's no cases where dasatinib alone will be sufficient. You need additional signaling axis to be blocked to actually have an impact on cell survival or tumor survival or tumor growth inhibition. And that's a recurring theme. We see that across all our PDX and organoid explorations. We've also done that for colorectal cancer and also for AML. We see that finding 2 drugs and sometimes 3 drugs is essential to actually inhibit cell proliferation. And that's clinically, that's a somewhat hard message, because if you would do this in the clinic, it would mean that you need to do clinical trials with multiple drugs. And multiple drugs means multiple pharmaceutical companies. And they don't have a real incentive to collaborate. So they always want to have mono treatment, or if they have 2 drugs available, single manufacturer makes 2 different drugs, then they are inclined to spend time on this. But we talked to clinicians and they said, well, this will be hard for clinical implementation, because clinical trials that show that it actually works, it will be very hard to be financed. So that's the thing we have to deal with. But I think in the context of pancreatic cancer, we've seen that sometimes 3 drug combinations work best. And then what's really important to note that these are always drug combinations where we use low concentrations of drug. Because if you start to use single drug concentrations and then combine it across multiple drugs, efficacy goes up, but also toxicology goes up. So it will be too toxic to give to patients. So what we then do is to reduce the dose. So toxicology profile is okay, is tolerated, but the individual concentrations of kinase inhibitors are much lower than you would use in monotherapy. And that actually works very well. So there is really an angle, there is really a window of opportunity for combining different kinase inhibitors at physiologically relevant concentrations.
Yeah.
Hello, Piers-Man. Thanks for your excellent presentation. I have some questions to ask you. I have used INCA to predict kinase activity. I download the resource code and run successful in Linux platform. So I can check carefully the output file. But in the results, I couldn't find the substrates corresponding to the highly activated kinase. Also, there is kinase and the substrates network file in the output. But is it available to find the interactional substrates for each activated kinase? So if I get this list, I can do the downstream analysis, for example, enrichment analysis.
Yeah. So you would actually, instead of having the kinase substrate plot, you would like to have the tabulated data as a text file, so that you can do the downstream analysis.
Yes.
Yourself. Yeah. Well, the easy way to actually get that is to contact Alex, the administrator of INCA, because he can provide you with that, no problem. So just contact him, and I think on the INCA website is contact details will be there. He's actually sitting here in the room next to me.
Okay, I know.
Yeah,
I
so
have another.
just contact him, and he will provide you with the data. For all these tools, you really have to apply it to your own data sets, and then you see how they actually respond. And then it's good to compare different tools, because then you can see they don't give every time the same results. So between that, I could show you that for the PTMC and KC app and INCA, that for the top scoring kinases, the top ranking kinases, you will get more or less the same pattern. But then if you go a little bit more down, there will be tool specific kinase activities, and it probably will take like a comparison of many different experiments to actually assess which tool is best. So that's also actually where we did not try to assess that in the review. We only made like a example comparison to have like a user experience. How hard is it to actually do the analysis with different tools? But we don't have a back-to-back benchmark of which tool is better than other tools, because I think we lack the ground truth data sets to actually do that. And frankly, I also don't think that the field is mature enough that you can really do like a back-to-back comparison and then a ranking of how that one tool is better than another tool. I don't think that's assessment can be made at this point in time, maybe in a few years. But I think the evolution will show that the use of tools it really reflects how convenient they are. So some of the codes that we tried to work really they were very hard to get them to run. And with INCA, that was easy because we do that ourselves, but also PTMC and also the KC app. Those are tools that everybody can use. They're user-friendly. The theoretical framework is reasonably well established, and they have developers that actually maintain the code. And those ingredients are all needed for a tool to be adopted and used by a wider community than only the developer or the development lab itself.
Okay, I have another question. As you see, the software is developing. I find an algorithm to predict kinase activity was published this year in Nature Communication called KSTAR, K-star. The K-star algorithm is based on the hypothesis that the more activity kinase is, the more of its substrates will be observed in a phosphoproteomics experiment. Which method do you think is more reliable for predicting kinase activity based on the quantitative values of the substrates or based on the count change of the substrates?
Yeah, we were just a little bit too early with the final version of our review, and then indeed the K-star was published. So I would have liked to include that, but at the moment that the editorial office was already making the proofs for our review, it could not be included anymore. Having said that, I think it's a very interesting implementation, and I'm not sure whether it's better or worse. I think it integrates a lot of the concepts that are already in the older tools, but I'm thinking, I don't think we've tried to use it on our own data yet, but that's on the to-do list. So we are very interested in how K-star performs. Yeah.
Okay.
But I don't have a preference for whether it would be better or less good than our tool or PTMC or KCApp. Yeah.
Okay, thank you, Prisma.
Thank you, Dr. Prisma. I'm quite impressed about your result between your INCA prediction and the result of the cellular experiment date. I have one question that in this experiment, the cellular date maybe because mainly rely on the human cell lines. But I have a question: if we have in the drug development, if you send our data for the IND, we always we usually need to provide the data from the animal model. What's your advice is if we conduct the animal model experiment with your INCA prediction?
Yeah, it's a good question. So it really relies on kinase substrate relationships, and phosphoside plus is richest for human, but you can. We are working in PDX models and also in genetically engineered mouse models. So PDX models are not that hard because it's just human in a different organism, but you are really looking at the human tumor proteins. So that's human material, human search. But once you have a genetically engineered mouse model, which is beautiful for drug discovery, you need to have really like a mouse version of INCA. And what Alex worked on was to take the orthologs of the human sequences of the phosphosides and then see which mouse orthologs are there, and then make a mouse version of INCA. That actually was not trivial, but we are quite far with that. But indeed, you need to have a kinase substrate relation database as well as a annotation of the genome or proteome for all the kinases before you can actually do any of the upstream, any of the kinase inference tools. So I think we've spent like 3 years to mousify INCA, and that was actually pretty complex work. So human is easy because we start from human, but then translating that to mouse or maybe even to rats or to other organisms that is really a hurdle to say, because you really have to change the code, or at least the input data. So it's not simple. It can be done if there's enough demand. I guess it will happen. But our experience with the mouse Inca was it was harder than we thought. But I can imagine that indeed from a preclinical drug screening idea in model organisms, it really makes sense to have the mouse version, or maybe even a yeast version that can be used. But I cannot oversee the difficulties yet. I know that for mouse it was already hard. So if you go further, I think for yeast, the relationships are much, all the data is already available, but you should translate it into the right format to actually run the code on it. So it can be done, but it's an effort.
Good. I'm looking forward. Looking forward. Looking forward.
Just ask.
One more
ask
question.
Alex.
Okay. One more question. I see in one of your slides. I see the comparison result between two different tools. I see the result is not quite the same. Do you think it is necessary to conduct do you think it's important to conduct different analysis with different tools and combine this result together and conduct the following analysis?
We saw the same thing, that the top ranking kinases are typically highly similar across tools, but then going down, there is tool-dependent rankings. And what we now tend to do is that we use Inca, because we use that by default, but we also use PTMC, because PTMC has the advantage, it can run on single samples, and that's for our work requirement, but it also maps not just to kinases, but already to integrated signaling pathways. And that makes it sometimes easier to find which signaling axis is actually being activated, because it already integrates beyond the kinase, multiple kinases, into a single signaling term. So PTMC is our other favorite tool. It's well maintained by the Broad Institute in Boston. So we already do dual tool use on the same data, and also to be sure that we don't miss out on kinase or signaling activities that's isolated Inca would not see. So I think you can spend a lot of time on using your data set on all available tools, but in the end it will confuse you. I'm pretty sure it's quite confusing. So our advice will be use one or two tools, but don't spend time on comparing your data with all tools, because there is no single truth based on a single data set. Maybe in 5 years' time, there will be relative tool comparisons with a proper ground truth data sets, because that's what you need. You need a data set which you exactly know what kinase activities are being perturbed, and then you can see how the tools predict and score that. But in the absence of that, it's a hard assessment to make at this point in time. Yeah, so use two tools. That would be my advice. Yeah.
May I ask another question?
Yeah.
So I see the data you show. Most of the kinase shown in the top are the tyrosine kinase. So can you give us some kind of
that's a very sharp observation. You're right, and that has a reason. We use both tyrosine enriched capture, so phosphotyrosine, the PTAR IP, and we use global IMAC enrichment. And what we see that for in an oncology context, where tyrosine phosphorylation is most of the time the highest level of signaling. So it's the cell surface receptor signaling that initiates the signaling cascades. That the tyrosine phosphorylation is most relevant for oncology. And so if you capture phosphotyrosine peptides by either the bTyr IP or with SH2 domain capture, then you immediately zoom into the relevant phosphopeptides and the relevant phosphosites that can also be mapped easily to activities.
Yeah.
Whereas if you use global enrichment, you get many more data points. So IMAC will give you easily 10,000 phosphosites, but it's only in in oncology context only how you call that? It doesn't really help you that much. It's all with downstream signaling instead of upstream signaling, and downstream signaling works well if you don't have tyrosine kinases. So, for instance, in melanoma, where you have how do you call it? Where it's more serine-threonine phosphorylation that's aberrant, then IMAC works okay, and tyrosine IP does not work. But for many of the kinases that we are looking at, it's actually tyrosine phosphorylation. It's tyrosine kinases, and they are by themselves tyrosine phosphorylated. So that's the most information-rich phosphopeptide subset. And we nowadays we combine it. We always do global and tyrosine phosphorylation. But when we started with Inca 4 years ago, we actually started with phosphotyrosine IPs, and that turned out to be much more productive than titanium oxide global phosphoproteomes. It's a good observation, but this is the background why you would still see that the most relevant signaling goes on the phosphotyrosine axis and not serine-threonine, not that much.
Yeah, thank you.
Yeah. Yeah. Other questions?
Is there any more questions?
No, the level of the questions, the quality was really good. You really, you know what we're doing, what we're, you understand how kinase activity inference, how it works. That's good.
One more question.
Sure.
Can this database or Inca tools used for the analysis of phosphohistidine analysis?
Yeah, yeah. I know it exists, at least in bacteria. In human, I'm not that convinced, but in principle, Inca and also other tools would be able to work with histidine phosphorylation, but I'm not sure whether the kinase substrate relationship databases contain any histidine phosphorylation at this point in time. I don't know. Maybe Phosphocyte Plus actually does, but so then it can be used. But I'm not convinced that in human cells it's physiologically of any relevance. But at the same time, I don't enrich for histidine phosphorylated peptides, so I wouldn't know. I think it's a part of the field that's a big question mark. We don't know how relevant it is, but it's really there, and what the physiological implications are. Yeah, but it's fascinating.
Thank you. Thank you.
Yeah.
Any more questions?
Let me ask a simple question, Sander.
Yeah, sure. I like simple questions.
Did you still remember that Alex maybe once presented results about a prediction of phosphopeptide and phosphoinsight from label-free DIA dataset that we generated together?
Yeah.
And could we use that prediction result as input to Inca and infer the kinase again?
Oh yeah.
If we can do that, I mean, maybe more label-free DIA dataset can do the kinase inference. Yeah, so maybe that will be a a great prospect, I guess, because I'm not in this field. I just think about.
I don't recall what the status of that is, but indeed you can indeed predict kinase substrate relationships, but they need to be really good, because otherwise you generate a lot of noise.
Yeah.
So the quality of the prediction or the filtering of the predictions should be really strict in order not to contaminate the activity inference. I guess it's best to contact Alex on what the status is from that, and whether it really would help. Yeah, it's yeah. Thanks.
Okay, now it's just time to wrap up. Before we wrap it up, our sincere gratitude goes to first and foremost the speakers and the panelists, and also wish to thank our team members behind the scenes, and of course, thank you our listeners and our audience for watching our webinar. And that's all for tonight. Thank you and bye everyone.
Thank you for all your good question and attention.
Bye-bye. Thank you.
Bye.
Well, thank you. Thank you.
Thank
Bye.
you. Bye.
It was really great.